Here we go with our first set of video notes for our Biology 1 class. I'm calling these notes Principles of Biology. We'll be discussing, obviously, principles of biology, so some of the elements of nature of science. So what does it mean to be a scientist and how does science work? Talk about some of the scientific method and how the scientific method works. And finally, we'll just get into a little bit about what biology is in a general sense. Remember, you're going to be taking notes with me as you go. If you're not sure what page in your notebook to be doing this on, you can check on the video description. You can also look on the website. It'll say what page you'll be using there as well. Don't forget to set up Cornell Notes on that note page, and each of the headings of each slide should be your key term that you write in the left-hand column. So to start off with, the nature of science. So what does it mean to do science? What does it mean to be a scientist? What are some of the elements of all science or problems that all scientists need to deal with? These are some things we'll be discussing now. So again, remember this right here is going to be your key term on the left-hand side. And feel free to pause at any point if you need to catch up or write something down. So, first major thing about science, at least the major thing that I focus on, is we're never 100% sure about anything. We never know we are correct. We can be pretty confident. We can be like 95% confident. We never know for sure that we're correct. And how can you never know you're correct? You know, you do a math problem and you feel like you know that you're correct. Well, in science, everything we do is affected by bias, right? Bias is how our life experiences affect our interpretations, right? So maybe being somebody from Dallas, you might see as what you do on the weekend as being something totally different from what somebody from New York City would see as being something you do on the weekend. That's your life experiences affecting how you interpret what a weekend should be like. The story is always changing because new evidence is always being found. Think how fast technology is changing. As we get more technology, scientists can do more stuff, right? Charles Darwin would have loved to know the human genome way back when he did his work, but he didn't know that. Now we have the entire map of all of our genes, all of our DNA is all mapped. So that's a huge leap forward. We can use that for making more discoveries. So as we make more discoveries, we might change what we thought was true before because we learned something new. And finally, as a result of never knowing we're correct, we always say that our hypotheses, what we do in an experiment, are supported. Okay? We never prove them, because to me, prove means we're 100% sure, right? So we could do an experiment and we could gather lots of evidence in support of our hypothesis, and we could be pretty confident that our hypothesis is correct, but we can never prove that it's correct. And it still could be something that we didn't see, something we don't know about yet. And the last thing is a theory. Is a little bit different from the way we use the word theory in everyday life. In science, a theory is a scientist's best possible explanation, right? It's extremely well supported, right? Like the theory of relativity or theory of evolution. They have lots and lots and lots and lots of evidence behind them. One person doesn't just come up with something and say, I have a theory that X, Y, or Z. A theory is something that's been maybe come up with somebody originally as their hypothesis, but then other scientists have researched that thoroughly and gathered lots and lots and lots of support for that. Okay, so moving on, you can pause here again if you need to catch up with anything, but we're going to move to the next slide. The scientific method. This is something you guys have probably learned many times. We do focus on it a lot in biology, trying to use these steps. These are guidelines, really, kind of like Pirates of the Caribbean, more like guidelines. You don't have to hit all the steps. Not every scientist hits every step each time, but they are a good sort of roadmap for how you can do science. So, that being said, the first step is pretty much always going to be observe. Okay, you observe something. All right, you observe that you smell smoke. Okay, the question then would trigger in your mind is there something burning? Right, so you have to make that observation to trigger that question. Right, questions are always triggered by some sort of observation. Then you would make a hypothesis. Right, a hypothesis is your prediction. Right, it's a prediction that can be tested, or you could think of it as your answer to your research question. This is often done in an if-then statement. So if X happens, then we will see Y outcome. We'll talk about this a little bit more later. Once you have your hypothesis, you design and conduct an experiment. So you collect data in a controlled way that allows you to gather evidence in support of your hypothesis or to possibly refute your hypothesis. Once you've done your experiment, you're going to take a look at all your data, maybe do some mathematical tests on it to see if things are different, and you decide, was your hypothesis supported? Remember, we're going to say support. We're not going to prove. So was your hypothesis supported? And then this last step is something that I like to add because I think it's really important. You'll never really see it in any textbook. So this is why this is sort of italics. This is Mr. Godkin's step 6. But share, right? You need to share the results of your work, no matter what. And scientists are always sharing. Scientists share their work in scientific meetings where lots of scientists come together. They share their work through printed journals, so they publish their results so other scientists can read them, right? Let's say we do an experiment and we look to see if garlic could cure cancer. And we find out that garlic doesn't really have any effect on cancer. Right, so was our hypothesis supported? No, our hypothesis was not supported. Should we still share that data even though we were unsuccessful? I argue yes. You should always share all your data because then maybe another scientific group then won't waste their time checking to see if garlic can cure cancer. So no matter what, you have to share your results so people know what's happening. Now, a quick little discussion on difference between observation and inference. You'll probably see this word inference more than once throughout the semester. An observation, probably something you're fairly familiar with. An observation is the use of one of your 5 senses to collect information. So you can see something you could smell something, you can touch something. Okay, all this stuff is going to give you information. In inference, how is an inference different from that? Well, an inference is a conclusion based on that evidence, and that inference is always subject to your bias, right, to how you're interpreting what you see, touch, feel, etc. So for example, you look at our comic over here, right, anthropologists are coming. An anthropologist is a scientist that studies cultures of people, right? These guys are obviously all running to hide their technology. Well, in 10 minutes, these anthropologists are going to come through these huts, and they're not going to see any of this technology, right? Their observation is going to be no technology. Right? Their inference is going to be that this is a primitive culture, right? Inference based on their bias. Lack of technology means that they're not very advanced. But obviously, like we said, you were never 100% sure. They didn't know that these guys ran and hit everything, and they actually are quite advanced, and you're just tricking them. But anyway. Okay, so on to different parts of a scientific experiment. Again, you're probably familiar with a lot of these, but it's been maybe a little bit of time since you've gone over them, so we'll just kind of hit on everything again. So first off, your control group or your control is your normal state, right? It's your baseline, what you're going to compare to. So if something's different than the control, then you can say there was an effect. Right? And then if anything that's in your experiment is not part of your control group, that means that it's part of your experimental group. This is the group that has one difference from the control group, one little thing, and you want to see if that one little thing is going to affect your experiment or affect your outcome. Right. Within your experimental group, you have a couple different variables. The independent variable is what you change, the one difference. If you have more than one independent variable, that's a bad thing, right? Because then you don't know what caused the observed results. Right? If you change temperature and humidity in your greenhouse and your plants start to do better, okay, great, they're doing better, but is it because of the humidity or is it because of the temperature? You don't know because you changed 2 different things. And then the dependent variable is going to be what you measure. Okay, this is the thing that you measure. So in that previous example, we would measure how well plants are doing or something like that. And whenever we're graphing these, the independent variable, the thing that you change, is going to go on the x-axis. The independent variable is going to go down here. And the dependent variable, what you measure, is going to go on the y-axis. Okay, so let's go through a brief little example of how we would use the scientific method. So first off, I'm going to make an observation that my DVD player, or whatever, my DVD isn't working. I want to watch a movie and there's no sound. So obviously the first question would be, why isn't this thing working? All right, could it be that maybe there's a bad connection in one of my wires? Who knows? So my hypothesis, my potential answer to that question is that, yeah, it is a bad wire. So my hypothesis in an if-then statement would be, if I fix the connection of the audio cables, then it will work. So I've got that in if-then format. It's a potential answer to my question of why it isn't working, or could it be a bad connection. All right. Our step then would be to do our experiment. Right? So my control, that's easy, is the non-working condition it's in right now. That's what I'm going to compare to. That's what I want to change in this case. My experimental group is going to be my changes to the wires. Okay? My independent variable is making sure that all the cables are plugged in well. Should I change anything else? Should I mess with the volume? Should I change any of the audio settings? No. Right? I should keep everything the same if I want to see if it truly is the cables that fix it. I could change all those things. My DVD might work, and that might be great, but that doesn't tell me what made my DVD not work. My dependent variable is what I'm measuring. So what am I measuring in this case? Whether or not I can hear my movie. All right, so I do all this, and now I can hear my movie. Awesome. So my conclusion then would be my hypothesis that a loose cable disabled the sound of my entertainment system is supported. Right? Maybe something else crazy was happening. Maybe there's some kind of electromagnetic interference that I don't know about that was causing that to not work. But the evidence points to the fact that, yeah, that works. And then finally, I'm going to share this. So I'm going to post on Facebook that I bought crappy audio cables that don't really seem to work all that well. And it's important to share that so that my friends don't go out and buy those same audio cables that for some reason seem to come undone a little bit easier. Okay, so moving on, talk about biology again. So biology is, well, bio is life, ology is the study of, so simply put, biology is the study of life. Yeah, that's not that difficult, but what does it mean to be alive? So the major science-y stuff that we're going to be learning about for this unit are the 6 main things that define life. Right? If you're going to be considered alive, you need to have or do each of these 6 things. Very important. So the first thing that all living things have are cells. All living things are made out of cells. They might just be one cell, a single cell, a very simple cell like a bacteria, but it's still made out of a cell. All living things have some sort of organization. They're organized. Now it doesn't mean that they have all their stuff in a sweet trapper keeper. That means that all their information is organized at a molecular and cellular level. All right, so this is usually in the form of DNA. Right, all of our genetic information is stored in DNA. DNA is the blueprint of life. There's also something called RNA. We'll talk about a little bit later on. Or you could see that different cells have different organelles, which we'll talk about again later on. They help organize what's going on inside the cell. But there's some level of organization. The next thing is something called metabolism. Metabolism, simply put, is using energy. Right? When you think about if somebody has a high metabolism, you probably heard this before, probably somebody who's really skinny because they use a lot of energy. They take their food and they turn it out and turn it into energy really, really quickly. They don't store it. That's metabolism. So all living things use energy. They need energy somehow. Another big term we're going to focus on is all living things maintain homeostasis. Right? Homeo means same, state means or stasis means state, so literally means the same state. So you do something to keep your body in the same state. Often, this is in response to the outside world, response to external stimuli, we sometimes say. So for example, if it's really hot, your body might start sweating to cool you off. Or vice versa, if it's really cold, your body might start shivering to warm you up. Those muscles move around using up energy. Right? The most classic example of homeostasis would be your body temperature. Right? We all maintain roughly the same body temperature, 98.6 degrees Fahrenheit. But this is just one of many, many, many examples of how we maintain homeostasis. Also, all organisms grow. Right? Same reason your jeans that you wore in 3rd grade don't fit you anymore, because you've been growing. And then all living things need to reproduce, because if you're not passing on and you're not making new critters, then what's the point of life? Right, so 1, 2, 3, 4, 5, 6 things you have to have all of these things to be considered alive. If you don't use energy, but you have all the other stuff, well, you probably wouldn't grow and reproduce if you didn't have energy. But whatever, you're not alive, right? If you don't reproduce, you're not alive. Now, does that mean if you don't live to reproduction or whatever, you're not alive? No, it just means like as a species, you're capable of reproduction. Okay, with that, we are all done.